Zach Pitluk from Paradigm4 telling us about some data science applications. So leveraging Allotrope format. So Zach, off to you.
Great. Well, thank you very much. I really appreciate the opportunity to present. I'll be doing a few slides, and then Srikant Sarangi, who's my colleague in the commercial world at Paradigm4, a pre-sales engineer, will do a demonstration of the software. And this all, the genesis was really in conversations with Amnon and Matthew Fox, showing them that we had the software implemented. So thanks again to Allotrope, and thanks for all the brilliant presentations preceding us. It's all really good, and very excited about the chromatography and column database that Dana and Steve talked about. So today I want to present how we can complement the ADF with a scalable analysis and analytics platform, which we've called Reveal Analytical Development. And so just a quick overview of the technology, Reveal Analytical Development, the data diversity, and how we manage the information that's in the ADF files. And then we'll get into the demo. So this is a mashup slide that was created by Amnon, which I really liked. And it really speaks to the fact that the data that's captured in the ADF is a set of matrices, right? And the metadata itself, of course, is represented in the graph format. But as anyone who knows something about matrix algebra knows, graphs are actually nicely represented in matrices as well. So it's really a tribute to this idea of starting with matrices and preserving them all the way through into the analytical platform. So the lab experiments as a matrix, the ADF files as multi-dimensional arrays, data cubes, the importing and mapping of the ADF data description metadata to SciDB schema, and then the analysis that's done in SciDB. So SciDB is the scientific database that was created by Mike Stonebreaker, where data is represented on disk as an n-dimensional array or a vector. So that's unique. And it's not stored as a file. It's not stored as a table, which you can think of as just as a 2D array. It's stored as an n-dimensional array where you use rather dimensions to substitute for indices. And this allows us to have an underlying column structure that scales to thousands or more columns in each data model in SciDB. So it's essentially enabling the representation of data as arrays without limits to scale. So the basic concept that we have is this idea that on the top layer you have the interfaces for data scientists, R and Python, and browsers for scientists that just want to browse data, connecting through APIs, either REST APIs or R API into SciDB, which then sits on some hardware, either in the cloud or on premises. And we have a whole suite of these. We're very prominent in the omics space, so UK Biobank and those types of data sets. The basic concepts I'll walk through in the next few slides just to kind of explain, well, what does it mean when you have data stored as an n-dimensional array? Why would you want to do that? And the basic answer is slicing and returning subsets of data. So what you see here is just, in the middle column is just really the variety of different data that we know can be stored in SciDB as arrays. And on the right side, you have an example data cube where I've shown the axes that you can slice along, right? So what this means is that your queries in R or Python or your GUI would be asking to return data, for instance, along the dimensions of samples project IDs, feature IDs, and features could be chemical entities, they could be projects, anything along that line, as well as normalization. So these axes, of course, would get created for the particular project, but they're generic in the sense that you are able to grab hypercubes of data that you would normally try to pull out individually from all of these different files. So with regard to the the metadata, it's an interesting challenge, and I think Wolfgang said something really smart in that last many things, but one thing in particular, which is for a particular analysis project, you'll really only need a subset of the metadata in order to construct an analysis query. And what we're showing here is simply how you might organize some of the metadata that's in the ADF file into a logical hierarchy of arrays. And think about this now as taking the graph elements that you see, the hubs and spokes, right, the nodes and edges, and beginning to project them now onto a matrix where you have representing an acyclic directed graph. There's good work that's written on this by Lincoln Labs, and Jeremy Kepner is one of the lead proponents of using matrices instead of the current triple store graph structures. So what this allows you to do then is to subset along the different axes. So for projects, obviously through different ontologies, and these could be experimental ontologies, these could be any type of ontology that you want, as well as other information that I've just shown as an example, like excipients or excipient lots, things of that nature that could be in the metadata. Pipelines that allow you to actually work with both raw and normalized data, which can be very helpful, especially if you're interested in intercomparing different instrument results where you don't want their normalization routine necessarily. And then, of course, the information for the features, which could be things like in-sheet, could be chemical names. There's analytical scientists use this word feature, but it's very flexible to essentially name the entity, right, of whatever it is that you're storing. And the beauty is that the data then is stored in a cube on disk that allows the data scientists to slice that data out in a routine and reproducible way so that they can filter their interest using these arrays, the metadata, and arrive at the values that they want. And so, with SciDB, we're not talking about like individual files worth of experiments, right? We're talking about wanting to aggregate 10 years of data or 3 years of data or a year of data even into SciDB. So we're really kind of at that next level up from where the other analytical platforms that I've been discussing. So we're going to show you the implementation in SciDB of the Agilent data set that was made available through ADF and through Merck. And it's a simple experiment, absorbance measurements of an 8-component mixture. There were 17 injections, 8 wavelengths were monitored. There was variation in the detector data rates, detector resolution, and run times, which comprised an instrument DOE. And so Srikant will show that in a minute. I just wanted to kind of further kind of explain where Paradigm 4 is going, and in 2 slides, just to put that into context. So this is the schema that we used in the design of experiment. So we have an array that allows you to slice along wavelengths, iterations, experiment number, signal info, and then in the data cube, you simply have the signal attributes. Clearly, SciDB is capable of doing more than that. And then the metadata array has 8 different attributes that you can use to filter. The there are a couple screenshots, but you'll see it live, so I'll just kind of scoot through these of R Shiny viewers. The whole thinking is that because everything's in R, if you need to set up a viewer or a GUI of some sort, it's very easy to do. The same with Python using Plotly, but we'll show you results with R Shiny. And the basic idea that Paradigm 4 is going after, right? Because we're really trying to, we're redefining some of the basic ideas in high performance computing is captured in this slide. So we have these reveal suite of apps that feed into SciDB as the primary cluster, where the data is highly organized and serves to enable you to do large elastic compute analyses using this burst mode capability. We have extensive examples in the area of genomics, but we're happy to talk to you about doing this and setting up POCs in the ADF format. And then we've also developed this flexible storage system, which is called FlexFS, which sits on top of S3. It's a file system that has POSIX compliance and scales linearly with the number of workers, which is why we designed it, because there's really nothing out there that allows you to do this kind of scaled analytics in the cloud. And then just to be explicit, where you would store the large data sets would be in Flex, as well as in the shared reference in S3, so for data that you wouldn't encounter very often. So you can easily see a nice hierarchy where you can quickly shift data into SciDB for large analytical experiments, but it would stay in a file store directory, which then could be easily addressed and accessed through other programs. The data here will be stored from SciDB as an arrow format to facilitate really rapid transfer of data from SciDB into Flex as well as into S3. And then finally, this really captures kind of the main concept of being able to then have multiple different SciDB clusters reading from this shared file storage, the chunked data, so that you're not transferring all the data, just the pieces that you're interested in. So we'll take advantage of this whole concept of chunking and then serving burst workers. And because we're using the arrow format, it's generic and can be used by any machine learning language. So with that, then I'm going to hand off to Srikant, and we're going to do a demo. So I guess we have to transfer the control to Srikant.
All right, just okay. So I think my screen should be coming through now.
Yes.
Yep. Okay. Awesome. Hi. So I am Srikant Sarangi. So I work as a pre-sales engineer with Zach, and what I would like to show is how SciDB works with the Allotrope data format. SciDB, as Zach showed in the presentation, is a computational engine under the Reveal Analytical Development application. The data that we will be using is the Agilent ChemStation LCUV absorbance data collected for performance testing. We got this data from the Allotrope Foundation, and many of you might already be familiar with this data, but briefly, the data set consists of absorbance measurements from an 8-component mixture, a number of variables that were altered to generate multiple combinations of data for the performance testing. These include detector rate, resolution, runtime, and the wavelength. And for each combination of the data, as you can see in this table, there were 17 injections that were run. So there were 17 ADF files for each sequence, amounts to 136 files, millions of data points, over 1,000 chromatographs, and the data footprint was roughly 80 gigabytes. So it's not a massive data set, but we felt it was a good size to show how we can work with the ADF. So using this metadata and the data cubes in the ADF, we wrote a prototype loader, used our SciDB to Python API to load the data into SciDB. All the data was ingested in approximately 10 minutes, and this is without any parallelization or compression, just a very basic implementation, of course, something we can optimize to run much faster. As you might recall from the slides that Zach had showed, all the metadata was read into one array with the various attributes, and the waveform data was read into another array. So in order to make the exploration of this data more user-friendly, as well as to highlight the interoperability of our system, we designed a simple GUI using R and R-Shiny. And this application talks to SciDB using the SciDB R API that we developed. So almost all the queries that I'll show are being done on the fly. There's no caching or indexing. I haven't run these queries before. So in this panel here, we see all the different parameters associated with the data. Now, as we make selections, these different waveforms, so the different wavelengths are the rows, they get updated and displayed with the different detector rates. These show up as the different columns. And so we found interfaces like these really allow user to quickly inspect and explore the data. And by hooking into SciDB as the underneath layer, such GUIs become really efficient in terms of time spent in extracting and displaying the data. Actually, some of the lag that you see is as R pulls in these data sets to actually plot. So, this is for demo purposes, obviously, but you see how as the wavelength goes up, the waveforms start breaking down. We could look at some few more. And one of the metrics that we thought that might be interesting, given that there are 17 injections for each parameter combination, was looking at the variability amongst the injections from a perspective of calibration of the instrument. So if everything was perfect, all the chromatograms would be identical. So that's clearly something that we wanted to check. So I'm just going to select one of these. Let's do one. And sorry about that. There are not all the combinations.
Some of the data is missing in some of these.
Right,
so the on-the-fly calculation can't work.
Right. So some of these combinations, for instance, 210 with the certain runtime. So it basically it tries to do a calibration on the fly, and so it's a good proof that none of these queries were actually run before. So what it's doing is it finds for one of the peaks, it does it calculates the peak height and that compares it against the average for the runs, and then displays the variability. So that's what it should be doing right now. All right, and this is just something that we thought might be useful for demo purposes, but obviously someone with more knowledge about this data might have some other methodologies or might be interested in some other forms of calibration. And then finally, another interesting question that applies to this data, given that it is evaluating performance of the instrument across multiple variables, was doing a design of experiment calculation to find the parameters that optimize separation of the peaks. So we wrote up a function to optimize peak separation. It takes a few minutes to run, so I pre-computed that. But this is essentially what it comes up as, that we see how the separation is one of the parameters that we find for a particular signal runtime and resolution. So with that, that was what I wanted to show in the demo. So I will hand it back to Zach if I can figure out how to do that. There you go.
That's okay. Okay, great. Thanks, Srikant. Really appreciate that. I think we'd really love to get another collaboration going. We're happy to share all of the work that we've done and to make available if it's of interest SciDB into Allotrope to begin pushing this forward a little more. We do have good connections with some cloud providers, so we may even be able to get some free storage and compute from them. But that's something that we want to propose and make sure that you understand this is a very collaborative effort. So, are there any questions?
Yeah, so thanks, Srikant and Zach. So then maybe I'll ask a question then of the speakers. So Zach and Srikant, in terms of the direction of travel of the Allotrope format and the offerings feeding into data science tools in general, maybe just take it a little more broadly. Any sort of insights for the Allotrope Foundation as we're proceeding forward along our journey to standardize data? What we could be thinking about to help the data science community in general?
Well, I mean, I think just the basic conception of what Allotrope's trying to do is just extremely valuable, right? The challenge is, and I think that you see this in so many of the talks today, is the nuances and the complexities of the different experimental designs that are implemented. And I think it makes a lot of sense for the first sets of projects to be in kind of the analytical development space, where the number of experimental techniques is more constrained because research is kind of all over the map. So it makes a lot of sense what you're doing. So we're just happy to participate and to help promote what you're doing and enable the scaling, right? I think it was Mike Kress who gave a talk about 3 years ago at Rahway when the Allotrope meeting was there, who really outlined the grand vision of being able to have access to years of data in order to do the simulations and be able to identify essentially hidden quality, critical quality attributes that are necessary in order to reduce OOS and OOT. So, we're on board.
Right, and just to touch on Zach's point was the fact that we do these matrix linear algebra calculations so effectively makes it really helpful to do QC over years of instrument data or experiment data, and that's where we feel that our system can actually be really helpful with the ADF format.
Right. So doing what Srikant means is like doing a principal component analysis on a matrix that's 10 million by 20,000 or a million, right?
Understood. Yeah, understood. And I think Jamie has also indicated, Zach, that perhaps another question was forwarded to you as well, if you can see the chat window. If not, Jamie, could you just read the question? I can't see the question. So.
Yeah, I can't see the question.
I can't see the question.
Sure, so I can just read it. So does data have to come to Paradigm in an ADF format, or are they converting data from instruments into ADF?
It would come from the ADF. So we had a nice experiment, a proof of concept experiment with Lawrence Berkeley National Labs, which was very early on in Paradigm 4's history, where we demonstrated we can actually read the data portion of the ADF. So in that case, it was an HDF5 file directly into SciDB without opening it into memory. And so that gives you the potential to import data at scale, because that was parallelized. We were working with the NERSC compute environment at Lawrence Berkeley. And so they had, I don't remember how many thousands of processors that we were reading simultaneously, but it was a 7 terabyte data set. So we're really designed to work with HDF5 file format, and the metadata is really partly a question of how much do you want to decorate, and but in terms of scale, there really is no limitation. Each one of those data cubes that I showed you can have millions of values inside of them. So it really is, fit for purpose for this kind of scientific data.
Great, thank you. Well, then, thank you, Srikant and Zach, for a great presentation, and thanks to all of our speakers for another really interesting session.